This is my video for my photosynthesis lab that's due this week. First of all, I just want to say that my papers aren't as pretty as they probably would be and have been in the past. I've been out of town for a work convention in Philadelphia, so I'm a little limited on resources, but I can explain more in the messaging part of it. So let's get started. Alright, so the first thing I want to talk about is the Calvin cycle. So I have it drawn up here, as you can see, and I have everything listed. So basically what happens is you have CO2 come in, and it goes from a phosphoglycerate, uses ADP to make a biphosphoglycerate, then it uses NADP to add a phosphate. So now we have a glyceraldehyde phosphate, and it takes that and it creates fatty acid sugars, amino acids, so it goes into the process of creating energy like it talks about in chapter 13. And then from that glyceride phosphate, we release the proton or the phosphate, and we end up with ribulose 5-phosphate. And then from there, using ADP, we get our ribulose 5-biphosphate, and then it just goes around and around and around. So that's kind of what the Calvin cycle looks like, to get better terms of it. Now, as far as light reactions go, it's a little more complicated, as you can see. So with the light reactions, light comes into the antenna complex. You've got your thylakoid space here, your stroma here, and your thylakoid membrane. So everything's happening along the thylakoid membrane. So you've got light coming into this antenna complex, and you have an enzyme splitting water into O2 and 4H+. And then you have, coming through that antenna complex, you have a phosphoplastoquinine, and then that also goes through here, the cytochrome BF complex. It floats across the thylakoid membrane and connects right here, and turns into the second antenna complex and turns into a plastokinin. More light's coming in. It travels through that antenna complex and attaches peroxidin to the bottom of the thylakoid membrane inside the stroma side, and then that peroxidin goes through and takes that H plus and NAD plus to make NHDP, and this is where the peroxidin NADH reductase happens. And then also from there you have H plus coming in to the stroma, and you have ATP being made here at the ATP synthase. Something else I want to point out is you have photosystem 1 here and 2, but I also made another drawing that kind of shows how the two work together, since again I was limited on resources on far as like my papers and stuff go. So what I did is I found this image in the book, and I think it's really good to show how the two work together. So you have light coming into a plant cell. You have into the chloroplast. You have it making ATP and releasing O2, taking in CO2, and then you have where it comes through the sugars. It metabolizes the sugars. Sorry, I know you can't see. Sorry, it metabolizes the sugars. And then it takes that into the mitochondria, and the citric acid cycle happens, and then oxidative phosphorylation happens. So O2 comes in, ATP's released. So this kind of shows you from the light reactions, the light coming in, how it goes through the process, and how it connects into the citric acid cycle, or the Calvin cycle, sorry, and what happens from there. So I just thought this image was really neat to kind of further explain the two, and how they interconnect with each other. So again, limited on resources, and I'm sorry about that. Next week's video will be a lot better as far as color and things go, but I hope I was able to explain the Calvin cycle and the light reactions enough for you. Have a good day.